Okay, it's our review session for exam 1. And first I had a question about the exam format, so I'll tell you that there will be a section of multiple choice with only one correct answer. And those are designed to really just test your knowledge, right? Do you know the material? It's just, if you know the answer, it will be easy. I will say that there are practice multiple choice tests on the Moodle site. And so if you want to practice, you can practice. I mean, those are not the questions that will be on the exam, but it'll be similar types of stuff. Okay? And you can take those multiple times because it should populate with 10 random questions from the test bank each time you do the test. And then, sorry, multiple choice, and then I'll have some short answer questions that are designed to be like half a page. You do not have to write complete sentences of drawing a diagram or making a table or something like that. As long as you can convey the answer to the question, that's fine. And then there'll be an essay question. The short answer, I'll give you some options. You can pick 3 out of 5 or something like that to answer. So if there's one that you really don't know, you don't have to do that. And the essay. And again, the essay doesn't have to, it's not really an essay, it's just a longer, more in-depth sort of concept question. And again, you can draw diagrams, however you can convey the answer to me is fine. Doesn't have to be complete sentences. Bullet points, whatever. All right, questions, that's what I'm here for. Go ahead.
So I just had a few things like just talking more about the IPSP and the EPSP, just like knowing the exact difference between them. I think that's stopping me.
Okay, so synaptic potentials. There's a question about synaptic potentials and the difference between EPSP and IPSP. So synaptic potentials are going to happen where in the neuron? Dendrites, right? Dendrites and soma, but dendrites we're going to say, the input. And neurotransmitter-gated ionotropic receptors is what is creating this. The neurotransmitter binds with an ion channel, and depending on what type of ion channel it is, it's going to depend on if we get an increase in voltage, which would be excitatory postsynaptic potential, or an EPSP, or if we get a decrease in the voltage, which would be an inhibitory postsynaptic potential, or an IPSP. Right? So EPSPs are going to occur when we have sodium coming in. So the forces are sodium wants to come in, potassium wants to leave. Right? And so when sodium moves in, that creates an EPSP.
So they're just ions?
Just ions.
Okay.
So the channel opens and sodium can flow in. Because it has a positive charge, that brings it from -70 up, right? There are 2 ways that we can get an IPSP. The first is we can open a potassium channel, and because potassium is concentrated inside the cell, it wants to leave. It wants to leave. And so if potassium with a positive charge exits the neuron, it's going to get more negative inside, so it's inhibitory. And then finally, the last type of IPSP, synaptic potential, is chloride, which is negative. And if the main chloride channel is a GABA channel, so GABA, the neurotransmitter GABA, activates the receptor, then chloride is going to come in, and that's going to make it more negative and inhibitory.
Is calcium an EPSP?
No. Calcium is a voltage-gated signal, right? So it's not a post-synaptic potential, right? That's the other part of this. So here we have a synapse here, right? This is pre, and this is post, right? Calcium's coming in the axon terminal as a signal for exocytosis. So it's not, has nothing to do with postsynaptic potentials.
So it stays in the terminal.
Yes. And it gets pumped out of the terminal back into gets pumped back up. So we good on synaptic potentials?
That makes more sense now.
Awesome, good. Brandon, you had a question?
Is there a difference between stem cells and neuroblasts?
Yes. So stem cells can become any cell in your body. A neuroblast can become any neuron.
Okay.
So they're similar, except one's just for neurons and one's for any cell in the body. So stem cells, you can grow skin cells, you can grow heart tissue, that sort of thing.
You said neuroblast, right?
Yes, the neuroblast is the precursor for any neuron in the central nervous system before it becomes differentiated.
And can you, I've been looking at the chart for like the couple of the peak of action potential and the falling phase. Can you like show like, it's confusing like when I look at it myself, can you like pull it up and explain that?
Mm-hmm. I'm just going to pull up the picture on the middle page.
So I printed it out, I just didn't know like.
So the question is, can I go over the action potential and voltage gated channels and this chart in particular? So again, we have EPSPs and IPSPs that are happening at the synapse, and only a little bit of those ions come through before the neurotransmitter releases, the channel closes, and then the pump is going to bring it either back to pump the sodium out or pump the potassium back in and bring the charge back to resting potential. If, however, we have enough excitatory potentials that can summate temporally or spatially and trigger the voltage-gated sodium channels to open, that's what the threshold is. It's just, if we can get enough voltage to hit -50, that's the trigger that opens the voltage-gated channels on the axon at the start of the axon, right? And so when that happens, so much sodium comes in that we go past 0 and it actually becomes more positive, positive 30, more positive inside the neuron. So that's the rising phase. As sodium's coming in and the voltage is going up, it goes past -40 and potassium channels open. The sodium channels close here, no more sodium can come in. But the potassium channels remain open. And so now potassium is leaving the axon. So the charge is coming back down in the falling phase because potassium is exiting. And when the potassium channels finally close, we're sitting at -75. Okay? So now, most of the potassium is outside the cell, most of the sodium is inside the cell, and we have to reverse that in order to have another action potential. And so sodium-potassium pumps use a lot of energy to push all the sodium out and pull all the potassium back in, And during that time, when the forces haven't built back up yet, we're in the refractory period. We can't have another action potential. Right? So when those sodium ions come in, those sodium ions flood in, they get trapped by the myelin and spread out. And as long as enough of them get to the next node to raise this from -70 up to -50, more voltage-gated sodium channels open, and sodium now floods in that node and spreads out. And so this is the saltatory conduction, where we're only regenerating at the nodes, and we don't have to have sodium channels and potassium channels and pumps built into the cell membrane anywhere there's myelin. We only need to make them and put them at the nodes, right?
Now, so you have the regenerating things there, right after the regenerating
That is the regenerating, right? So much sodium comes in here that enough gets down here and triggers a new action potential here with the rising phase and the falling phase. And that then spreads down. So that's the regeneration. At each node we regenerate a new action potential. Until finally we reach the axon terminal. So finally, all that sodium from the last node comes into here, makes it positive enough for calcium channels to open. Calcium comes in and activates synaptotagmin. So we have action potentials at the nodes. When we get to the terminal, voltage-gated calcium channels allow calcium influx. Snare is already holding the vesicle in place, waiting for calcium. And calcium activates synaptotagmin. The vesicle fuses with the cell membrane, dumps the neurotransmitter, and then is recycled and refilled. And then we have 3 ways that we get rid of the neurotransmitter so we can have another signal. So that's pretty much everything on this. Right? Yes, Sam.
During the migration phase of neuron development, so what are the 2 ways he said they could migrate in?
Yeah. So the question is, what are the 2 ways neurons can migrate during the migration phase of development? The first is that they crawl along radial glial cells that radiate from the inner ventricular zone out to where the cortex is being built. Right? Because remember, we're just adding more and more neurons to the cortex until we've added so many that it starts to form the gyri and sulci, right? So they radiate out on the radial glial cells, and then they release from the radial glial cells and follow a chemical signal with that sort of Philopodia, like foot growth cone that's searching for the right chemical. And they follow that chemical to their final destination. So radial glial cells, and then the chemicals are neurotrophins. Then they follow the neurotrophins to their final place.
Do we have to know cranial nerve function since we learned the anatomy in lab?
Don't think there's any questions on that. I mean, in general, yes, I would like you to know the function of the cranial nerves, but I don't think I asked that as a question.
neurotransmitter? Is there like a locking key, right?
Yes. So that's this chemical selectivity, right? They have to fit in receptors. So GABA will fit in a GABA receptor, but GABA will not fit in a glutamate receptor. Right?
Thank you.
Mhm.
This is on the slide showed yesterday. What is diastasis?
Diastasis, yeah. So that's where you can have stress play a negative role in allowing synaptic sprouting, right, and allowing that to happen. I kind of skipped over that. That's not as important.
Okay.
High affinity plus high affinity efficiency. I think that's it wrong. That's, yeah. Like, that means strong signal, right?
Yes.
Okay.
Right? So affinity means how tightly is the neurotransmitter attracted to its receptor, right? So sort of how long is it going to stay? Is it going to activate it and immediately pop off, or is it going to activate it and stay a little while, right? And then the efficacy is how effective is it at, when it's there, is it really stimulating that receptor, or is it barely stimulating the receptor?
So when it says that new synapses are produced, does that mean that there are like new clefts being made, or new branches of axons, or what does that actually mean? When it says like there are you're forming new synapses.
So I talked about that in a lot of different contexts. I talked about it in terms of throughout your life, talked about it during development. What are you?
I think throughout life.
Throughout life, yeah. So every time that you form a new memory, right, that memory is represented by new connections that have strengthened and formed between neurons in your brain. So we're constantly creating new connections, and the more you use a particular circuit, the stronger it gets, the more connectivity it has. And we'll talk about that. You'll see that in an application when we talk about learning and memory and neural networks and how neural networks are formed. Yes?
Can you explain how the phantom limb thing works again?
Yeah. So the question's about the phantom limb pain. And so because we have that homunculus, because this part of the brain is dedicated to incoming sensory information from your arm, right? If we remove your arm, it no longer gets its input. But what does happen is over time, the adjacent areas take over that cortex. So just like in this example, here, if we remove the middle finger, the third digit, the adjacent areas from the fourth and the second digit are going to take over that cortex. And now that cortex is going to respond to stimulation by the fourth and second finger when it used to respond to the third. Okay? So if you remove a whole limb, there's a much bigger area of cortex that gets taken over. And so the areas like the face come in because they're adjacent on one side, and the shoulder or trunk of the body come in on the other side. And they start this, if you touch the face now, it's stimulating the same cortex that the arm used to stimulate. And so until our brain figures out that, hey, that stimulation is not my arm, I don't have an arm, it's actually face area now, I have to relearn sort of that when that area of my brain gets active, it's not my arm causing that activity, it's my face or my shoulder. And that disconnect, that period where the brain's still trying to figure out why it's feeling the arm when you see that the arm's not there, becomes pain. Most of the time when our brain really trusts our visual system. And anytime our visual system can conflicts with other parts of the brain, it usually results in pain or discomfort. Like when you're reading a book in a car and you get car sick, right? Your eyes are saying you're not moving, but you are moving around, and your vestibular system is saying, yeah, you're moving all around, and that conflict that happens is what causes you to feel sick, right? So anytime you have sort of conflict, the brain doesn't know how to interpret it, so it tries to get you to take action by making you sick or causing pain. Other questions?
Can you talk about the 6 groups or classes of neurotransmitters?
Yeah. So the 6 groups of neurotransmitters are organized by how the neurotransmitter is synthesized. So acetylcholine is the only neurotransmitter that's made with choline and acetyl coenzyme A, so it's its own class. Okay. The monoamines, or the catecholamines, all originate from tyrosine, and the dolamines all originate from tryptophan. Okay. The amino acids are constructed, and are similar in terms of their how they're made, how they're synthesized. ATP is a separate chemical that can also be used as a signaling chemical. The neuropeptides are like the amino acids. They're a separate class, but they come from different nutrients that we ingest that are modified by enzymes in different ways, but they all share similar properties in how they're made. So they're all, and these are smaller in size, these are bigger in size, that doesn't really matter, but that's why they're all grouped together. And then gases is the last class.
That's the newest class, right?
Yes, that's the newly identified class of. There's neurotransmitters that have been identified that we didn't know about since then, but not a different class.
Mm-hmm. So is upregulation like mean the same thing as supersensitivity?
Yes, supersensitivity is what happens after a neuron upregulates its receptors.
I was wondering if you had any general study advice, like what students have said has worked well for them, whether it's writing more narratives, flashcards, Quizlets, anything like that.
Yeah, you certainly want to, neural signal is a big thing that we covered, and so you certainly want to make sure that if you didn't understand things in your narrative that I pointed out, that you do understand that now, right? The review points, I think, cover the main concepts. And so certainly I would work on making sure you know that information, right? I treat the textbook as more of a reference material for you guys to help you initially understand sort of basically what's going on in that section, and then I'm going to elaborate on it in lecture. Right? So I really kind of focus on the lecture and the review points as the focal point of the information I really want you to know, as opposed to like if there's something I didn't talk about in class that's in the textbook, which I don't know that there is in this section, it's probably not that important. And as far as like how to study, I think you should study however you know you study best. So if you know that making flashcards works for you, then do that. If you know that rewriting your notes helps cement that information, then do that. You know, different people learn in different ways, and so that's really going to depend on what your learning method is.
So in the review points, you mentioned that neurotrophins are involved in like 4 different roles. Can you go over like how they're all different, like what they're involved in?
Sure. Yeah. So here's a review point that Brandon's talking about. How are neurotrophins used in neural survival, developmental guidance, axonal pruning, and apoptosis? So for neural survival, I mean that kind of goes with apoptosis, right? So when a neuron is migrating, let me start. When a neuron's migrating, we already talked about the radial glial cells, but when it lets go of the radial glial cell, the developmental guidance, that's where it's being guided to its final destination by neurotrophins. So they're the signal for in migration once the radial glial cells are done with their job. Okay? When it gets to the place, if it gets to the right place and it becomes the right, it differentiates, right? And differentiation, should have up here, but the neurotrophins are what causes it to differentiate into the type of neuron that it needs to be, and if it does that correctly, then it's going to survive based on the interaction with the neurotrophins. However, if it doesn't have neurotrophin support, so a lack of the appropriate neurotrophin support is going to lead to apoptosis, programmed cell death. Finally, once everything is kind of settled, neurotrophins promote synaptogenesis. They promote we form way too many synapses and then we prune back to just the ones that we really want. And that process is done through neurotrophins. So if a synapse, you could be the right neuron at the right place, but you have 5 synapses. 4 of the 5 don't have the right neurotrophic support, only 1 does, so that becomes the one that remains. Any other questions? And I'll be checking my email this weekend. If y'all have a question, feel free to shoot it to me. I'll point you in the right direction. Sam?
Can we go over the cholinergic system, the serotonin system, like that kind of system stuff?
Yeah.
I don't remember.
You're talking about these slides?
Yeah.
So essentially what I'd like you to know about this information is what is the source for that neurotransmitter, where's it coming from, and where is it going? And if I tell you what it's doing, right? So for dopamine, there are 2 sources. There's the substantia nigra that has a pathway to the basal ganglia, which is involved with movement. Okay. There's a second source called the ventral tegmental area, the VTA. And the VTA sends dopamine to the reward system in the nucleus of accumbens and the frontal lobe for helping with concentration and stimulating activity in the frontal lobe, cognitive activity. The cholinergic system is acetylcholine, and it also originates from a nucleus in the pons and midbrain, and it goes everywhere in the brain, and it is for general arousal. So it goes everywhere in the brain, is for general arousal. Norepinephrine originates in the locus ceruleus, and it also goes everywhere in the brain, and it's sort of for like hyperarousal, hypervigilance. So acetylcholine is sort of you're awake and functioning. Norepinephrine is you're excited and the brain's working really hard. And then finally, serotonin originates in the raphe nucleus, and it is sent everywhere in the brain. And it's also sort of neuromodulatory, and it's more relaxation or decrease in activity. It plays some specific roles that we'll get into in the hypothalamus with hunger and satiety. It acts on emotion in the limbic system here. Is that good? All right. Anything else? I think you guys are well equipped. So, I will, spoiler alert, when we talk about sleep and dreaming and memory, you need sleep in order to form memories. So, it's important to sleep when you're studying. REM sleep in particular helps us consolidate information in our brain. We know that for sure. So, if you don't sleep, it's harder to form memories. You're working against yourself there. So,
Thank you.
yeah.